Hi, my name is Leanne Melbourne and I'm a lecturer here in Earth Sciences. My topic is marine micropaleontology. This course is offered to Year 3 and Year 4 students. So what is micropaleontology? Micropaleontology is the study of microfossils, anything to do with looking through a microscope. We focus on the marine environment, and in this course we will look at different marine fossil groups, how their skeletons and shells record environmental information, and how we can use this and their presence in the fossil record as proxies to reconstruct past climates. Using our knowledge of the past, we can also assess and predict how these organisms will fare in the future under ocean warming and acidification. So we can focus on plankton, tiny marine organisms. In the course, we mainly look at four main groups: foraminifera, coccolithophores, radiolarians, and diatoms. And they can be split into two ways, so by what their skeleton is made up of. In the case of foraminifera and coccolithophores, this is calcium carbonate, calcareous, whereas for radiolarians and diatoms, they are made up of silica, silicious. They can also be split by what type of plankton they are. So coccolithophores and diatoms are phytoplankton, tiny plants, algae that photosynthesize. Foraminifera and radiolarians are the zooplankton, which are tiny animals that consume phytoplankton, or also can be symbionts, such in the case of foraminifera, where there is a mutualistic relationship with forams providing a home for the phytoplankton in their spines, while the phytoplankton provide nutrients through photosynthesis and create an optimum environment for growth. We can also use these organisms to reconstruct things like past temperatures, the biological pump, the export of carbon from the surface ocean to the sediments, and sea ice distribution, determining glacial interglacial cycles. So how do we collect these samples? Here's my crude animation. We have planktonic groups living in the top part of the water column. We also do have benthic species of diatoms and forams that live on or within the seafloor sediment. But in this animation, I'm focusing on the plantics. So how they grow, their distribution, where we find them, and their abundance are all indicators of the environment they're living in. When they die, they sink to the seafloor. Over time, layers of sediment are formed. And then by taking a core sample from our ship, we get a snapshot into the environmental history. So by looking at the fossil assemblages through the core and knowing the relationship between the environment and these organisms, we can make predictions on what the past climate was like. So here is our colleague David Nafs from chemistry looking at a sediment core. When you get a core, it's split into half, so we have double the amount of sample, and then you can split the samples into different time fractions so that you can analyse the composition of microfossils at that time period to get an insight into the environment at that time. So what can we actually analyse? Here I'm using foraminiferas as an example. So we can look at species composition. So the different species have different optimal conditions. One variable is temperature, and so understanding what types of species are in our samples can give us an understanding of the environment. We can also analyse size. Within a species, maximum size is usually a sign of optimal growth conditions, so we can infer about temperatures and nutrient availability. We can also analyse shape. Some species have different shapes and morphologies based on the temperature. And finally, geochemistry. Analysing what the shells are made up of, we can link this to environmental variables such as temperature, sea ice coverage, and carbonate concentration. So firstly, species composition. Here we have different foraminiferous species and where you're most likely to find them in the ocean based on temperature. So the wider the colour plot is, the more abundant the species is in that area. So we can see that this is split into different biogeographic zones based on temperature preference. So if we have a sample dominated with Neoglobal Quadrina pachyderma, we can see that the widest part of the blue bar is in the colder temperatures, and so your sample is probably from the polar region, as this species favours colder temperatures. Whereas something dominated with Globigerina monadi will be from a tropical region and as warmer temperatures are favoured, and we can see that the wider part of the red bar is towards the warmer temperatures. It's important to note though that as foraminifera are passive drifters, they are found in all oceans, but as you can see with something like Pachyderma, it's just that the abundance is in the higher polar regions as they are polar species. Next, size. All species have thermal optimums. This is where the temperature is right for maximum growth. If we look at size, we can see that different species have different thermal optimums. Globerotala monadi increase in size with temperature, whereas something like Oberlina universa has two maximums, one at 10 degrees and another at 30 degrees. So by known optimum conditions for the different species means that when analysing fossil assemblages, we have a better understanding of the type of environments these organisms lived in, for example, whether they are optimal environments or not. We can also assess shape. This species, Globerotala truncatilonoides, has a different shape depending on the temperature. So therefore, by looking at shape morphology, we can have indication on temperature. For this species, colder temperatures cause a more lens-like shape, whereas in tropical regions, we see a more cone-like shape. So looking at the differences between the different types of shapes can give us an indication of the temperatures these organisms are growing in. Finally, geochemistry. Shells are made up of calcium carbonate, and within calcium carbonate there's a certain amount of magnesium that's incorporated. This incorporation is based on temperature. So by analysing the magnesium in the shell, we can also look at temperature. It is important to note, though, that different species incorporate magnesium differently, so they have different concentrations. So your equations are species specific. Understanding species composition, which species are found where, and how they grow under different environmental conditions, enables us to be able to understand past climates and predict how these species will fare in the future. We can do simple things by assessing the assemblages, abundances, and size to predict where samples come from. This is a sort of exercise we cover in practicals. We look at the samples under the microscope and reconstruct past climates and things like temperature. So pretend you have looked at these three samples under a microscope, and I want you to match the sample to the biogeographic zone. So firstly, you need to think about the size and also the species composition that we find in each samples. So if we had Globerotala monadi, this is a species that favours warm temperatures, whereas Neoglobo quadrina pachyderma is a species that favours colder temperatures. And then we also think about size. So the samples with pachyderma are a lot smaller, and pachyderma is a species that does form smaller sizes, and smaller sizes are indicative of colder regions, and so we would assume that this would be the polar sample. Whereas a sample that contains Globerotala monadi, which favours warmer temperatures, these are large specimens, large species, and larger forms are found generally in warmer climates, we can assume this to be the tropical species. So this leaves the middle as the temperate sample. If we had more time, we would identify the samples in this temperate sample to find species that are indicative of temperate subtropical waters. So I hope that gives you an insight into what we do in marine micropaleontology. Ultimately, it is using marine microfossils to uncover the past to help understand our future.